Recall that myofibers, muscle cells, are comprised of myofibrils, which are sort of little organizations of these proteins that make up muscle. And when we look at muscle tissue, there's a variety of different proteins that are of importance for understanding muscle physiology. The first is myosin. Right? So myosin is a filamentous and globular protein. So it has a filamentous portion, and then it has a globular portion, or a big head on it. And making up a myosin filament are two sort of myosin proteins. So it has two heads, and then this tail is braided, right? So you have a myosin head, the myosin tail, and then a very specific region right here, which is known as the hinge region. So I'm going to write hinge there. We'll just go ahead and write head here so that you know it, and then our braided tail, like so. Okay, so myosin filaments will combine together so that they make up something known as the thick filament. Okay, they make up the thick filament. So think of multiples of these combining together to make up this big, long, thick filament. Okay? Other protein that is of importance is one known as actin. And actin contains what we call G-actin, right, which is globular actin, but that then combines on something known as nebulin to make what we call F-actin. So what happens is you have multiple actin molecules sort of combined together like so, and they make this filamentous actin protein, and then you'll have others sort of wrap like this, and they all sort of center around a midline, right? And that midline is nebulin. Okay? So then these actin molecules, right, will make up what we call the thin filament. Thin filament. And the thin filament, like so, right, has a protein that is a filamentous protein that wraps around that thin filament, like so, right? And this protein is known as tropomyosin. Tropomyosin. Okay. Then attached to tropomyosin is a trimeric protein, meaning it has 3 subunits to it, right? And that is known as troponin. Okay, known as troponin. The other proteins that you should be concerned with are titin, and titin is an elastic filament, so it's what gives skeletal muscle the elasticity. Okay? And so it is a filamentous protein that sort of looks like this, like so. Okay? And then the fourth protein, that big protein that's important, let's go ahead and do this one in green. We don't have green yet. This is one known as dystrophin. And dystrophin is a filamentous protein, like so, that is involved in anchoring. So this is an anchoring protein. Okay? So now, right, now that we know our proteins, okay, we can start to organize them and how they're organized inside of a myofibril. Okay, so everything is sort of centered around something known as an M line. It's made up of a protein called myomycin, right? But this is our M line, like so. All right. Now, the thick filaments align on that M line. So you have a thick filament, like so. Thick filament, like so. Thick filament, like so. And then they're aligned, right? So you've got one this way, like so. And the way that they're aligned, right, is that you have your globular heads out here at the periphery, away from the M line. And then on this side, they'll be over here. Like so. Okay? Then what you have is overlapping that, right? You've got your thin filament, like this. And then same thing on the other side. Like so. And those thin filaments are connected to something known as the Z disc. Like so. So this is a Z disc. This here is a Z disc. Right, then remember, in the center we had our M line, like so. All right, now the region from Z disc to Z disc, this region here from Z disc to Z disc, this is what we refer to as a sarcomere. And a sarcomere is the functional unit of the skeletal muscle. Okay? So, then what we have is we have, remember, our titin. This is our elastic filament. Okay, and it runs from the M line to the Z disc, like this. So remember, this is what's going to give our cell the elasticity. Like so, right? That's our elastin. And then if we draw our cell membrane, draw that right here, okay? There is the dystrophin, which is green, okay? And that is going to anchor a thin filament. So I'm going to draw another thin filament down here. That dystrophin is going to anchor that thin filament to the plasma membrane. So nice anchoring of that to the plasma membrane. Okay, so then what we can do is we can look at the anatomy of the sarcomere, and we can pretend that if we shine a light through this sort of sarcomere, like we do in a microscope, what we're going to see is some different banding patterns, right? This is what gives up those striations. So this sort of region here, where we only have this thin filament, right? Notice that we only have thin filament there. This is known as the I band, right? And so over here, you would have a thin filament like this, right? And then over here, you would have your thick filament like this, right? So you have your I band is the region where there is only that thin filament, okay? Now, the region where you have overlap between the thick and the thin filament, that's called the A band, right? That's your A band. But some people will say that the A band is the entire length of that thick filament. Okay, that's the common one, right? So the A band is the whole length of that thick filament. Okay, we have our M line here, and then where we have only thick filament, right? So from here to here, this is known as the H zone. Okay, and so remember, right, that this sort of repeats end over end over end the entire length of the muscle. So if we shine a light at it, right, we get sort of no light coming through, medium light coming through, no light coming through, lots of light coming through, and then it repeats. And because of that, right, you get areas of dark, medium, dark, light, right? And that's what gives the striated appearance of that muscle.